So we mentioned that there are a few more or less independent ways that we might choose to investigate the brain. One way is through the study of pathology, that is, the study of cases of illness, disease, trauma, or other forms of change to the brain that result in some overt symptoms. When things break down, it's not always easy to say exactly what has broken down. And pathology, as a source of information, has some vexing characteristics. It can provide information that is otherwise absolutely unobtainable. We don't do experiments by cutting out parts of people's brain or destroying them, but life has a way of doing that for us. But when you come upon any case of a damaged brain, it's an individual case. These are not controlled experiments, and there's no guarantee that the kind of damage done is the right kind of damage to inform your understanding. Pathologies are very messy, they are uncontrolled, and the manner in which you interpret a pathology reveals as much about your own presumptions about the system you're looking at as it does about the system itself. So we're drawing inference about a damaged system, and that's always dangerous because what we're trying to figure out is what the non-damaged system is doing. Now, the literature here is enormous, of course, so we're just going to look briefly at one case, and it's the most famous case in the history of neuroscience. You may have heard of this. This is Phineas Gage. Phineas Gage, whose skull is shown very dramatically there. He was working on the railroad in 1848 in Vermont in the United States. And what they were doing was breaking rocks, quarrying. And to do this, what they used to do was drill a hole, put in gunpowder, tamp it down with a rod called a tamping iron, then lay a fuse, stand back, ignite, boom. But on this occasion, on the 13th of September, Phineas was tamping down one such charge of gunpowder when it created a spark. Health and safety regulations were not very good at the time. This ignited the gunpowder and fired the rod out of the hole and into his head. It went in through just above his left eye and emerged at the top of his head, landing several feet behind him. He was knocked over, and we're not sure whether he lost consciousness or not. Certainly he was conscious shortly afterwards. Doctors were called, and despite the massive trauma that he had just done, he had actually basically wiped out the front left part of his frontal lobe in the brain. He recovered in a sense. He was hospitalized. He was brought to the attention of a lot of doctors in Boston particularly. And the case became a cause célèbre, which is why we still have the skull around with us. And it became so infamous because of the changes that occurred to Phineas Gage afterwards. So he got this damage, and there was a change in him. That's the tamping iron in question, on display, I think, in Yale University. So he tried to go back to work in 1849, but something fundamental had changed in Phineas Gage. What we loosely describe as his personality had changed. Now, there's been a lot of myth and lore has grown up around this case over the years, and in fact the case is itself the study of historians as well as neuroscientists. But basically the way the change has been described is that he went from being capable, efficient, competent, well-balanced and friendly to being crude, profane, very difficult to get along with, impatient, liable to fly into a temper. And so his friends sort of said he was no longer Gage. This association of a change in a word we don't know what to do with, personality, that could be located, whose origin lay in a trauma to the brain, to a specific part of the brain. That was what captured the doctor's imagination, and that was the beginning of the modern neuroscientific link in which something as ill-defined as personality is once more associated with a particular part of the brain. This is Phineas Gage posing for a photograph afterwards, in a photograph that only became available a few years ago. You can see he's missing an eye. The later story of Phineas Gage is worth looking into. All kinds of rumors abounded that he had run away to Mexico and joined a freak show and so on. What probably happened is he probably moved on and got other work elsewhere, and who knows what his personality, how his personality developed in the ensuing years. Historical interest right now. But this was a very important case because it led to this association of the vague notion of personality with the left frontal lobe. Insights from this gave rise to some horrendous practices. This kind of association was the motivating reason behind the development of a surgical technique known as lobotomy, which was used to handle severely behaviorally challenged patients. In a lobotomy, basically the mechanics of this we replayed by inserting a surgical tool through the orbit of the eye and wiggling it around, turning the front of the brain into mush, after which people were reduced to compliant vegetables. This was a practice that went on for decades. Dreadful. As you can see, insights in neuroscience carry a specific charge. We have such strong feelings about ourselves, and we associate so many of those with the brain, that any way of framing the brain, interpreting the brain, or acting on the brain comes with very strong ethical considerations. Now, we're not going to dwell on pathology anymore. We also mentioned that animal studies are of some use, and we'll just say a brief word of caution about that. Nervous systems share properties across species. A basic principle of operation of the simplest nervous system is very similar to that of the most complex. Furthermore, nervous systems don't all come with brains. There are distributed nerve nets in very simple animals like hydra and jellyfish. And then as nervous systems get more complex, you find clumping together of nerves, creating first ganglia and then ultimately brains. And then brains have evolved very, very slowly, so that we can identify the outer layers of the brain are evolutionarily more recent than the deep-lying structures. So, for example, deep in our brain, we'll find a bunch of structures known as the basal ganglia. And if you trace the evolutionary line back, they appeared around the same time as the last common ancestor of the human and the frog. That's a long way back. The neocortex, which is the outside of the brain that we're looking at, that we notice is freakishly large in humans, has this wrinkly structure. That's a recent addition. All mammals will have a neocortex, but humans have more than most. But the basic structures are preserved from one species to the next with minor modifications. And when we come to look at animals very similar to ourselves, like apes, there are essentially no structural differences between ape brains and human brains. The relative size of the frontal lobe is an issue. There's no cell type found in ape brains that's not also found in human brains, and that applies down to different kinds of neurons. And we'll be looking at this in a little bit in a later video. So when you look at the human brain and you want to compare it to animal brains, there's a lot to compare. The only really uniquely feature that human brains have, unique feature, is the relative size of the frontal lobe. So what questions could you ask then by studying animal brains? You can study rat brains, cat brains, monkey brains, ape brains, not so much. We have similar concerns these days about studying ape brains as we do about studying human brains. But we come to the brain with very many questions. Supposing, for example, we were interested in following the mechanics underlying the mechanics of smooth eye pursuit, a phenomenon we met in the videos on perception. Well, that's something that we see in humans and something we see in animals. Now, there are going to be differences. Different animals have different integration of the eye into the musculature of the body, for example. But that seems like something you could usefully look at in animals. What about schizophrenia? Schizophrenia is not a simple thing. Schizophrenia belongs in the class of poorly defined mental disorders. There are very, very many views on what schizophrenia is. But one thing is very clear. Schizophrenia is a human disorder. It lies in the human social world. It lies in difficulties in integration into the human social world. And it doesn't make a lot of sense to ask about cat schizophrenia. There have been studies in animals that sought to inform us about schizophrenia, but I want you to recognize that it is quite problematic to transfer something which is so intrinsically human onto an animal projection screen. Reasoning, often held out as distinctively human, although there's so much evidence of different forms of reasoning in various animal species. That's a very antique philosophical view now. But certainly you can't understand reasoning except from a human point of view that is informed by all this old philosophical baggage. So there's no point whatsoever in trying to approach animal brains and look to inform yourself about reasoning. So as you can see, the questions that you can ask of brains depend on what you expect to find there and depends on the degree to which you think this must have an organic substratum. I put memory at the bottom there. I am not going to unpack that. Memory is a Pandora's box. Memory is not a simple phenomenon. The manner in which we characterize memory is absolutely unsettled. There are hundreds of psychological theories of memory out there. There's reasons to question every single one of those. When we then go to an animal and we ask about memory, we have to redefine the subject. So you have to redefine memory so that you can find something that is plausibly a real part of the animal's lived world. So there's huge challenges in taking these concepts that we develop for humans and applying them to animals. We can learn some things from animals, but not everything.